Hi everyone, it's Miss Lopez. This is the chapter 6.3 and 6.4 notes on fermentation and alternative pathways for cellular respiration. In the first part of these notes, we talked about the breakdown of glucose and the processes involved with that. In these set of notes for 6.3, we're going to start by talking about what happens when oxygen is not present and other types of cellular respiration that can harvest energy. Before we do that, there's a very important topic or piece of this puzzle that we need to emphasize, and that is the role of NADH and NAD+ in these reactions. We covered the role of NAD+ and NADH as coenzymes in the very first set of notes for this chapter, but it doesn't hurt to review them one more time. NAD+ is capable of taking electrons away from molecules. Now remember that when an electron is taken, a hydrogen is taken as well. This process is called reduction. So you can see how NAD+ is reduced while another molecule is going to be oxidized. In this role, NAD+ is reduced to NADH as an electron carrier. And then we saw how NADH can be oxidized back into NAD+. At the electron transport chain, NADH donates the electron and is oxidized to NAD+. And the electron goes on and it creates the chemoosmotic gradient that is needed to produce ATP in oxidative phosphorylation. So in that case, that very first transmembrane enzyme that is embedded in that inner mitochondrial membrane called NADH dehydrogenase is reduced when the NADH donates that electron to it. As long as we can keep this cycle going, as long as NADH is being oxidized back to NAD+, and NAD+ is being reduced to NADH, this very important step will allow chemical reactions to take place. So please understand that this this oxidation of NADH back to NAD+ is critical to allow reactions to continue and the production of ATP to happen. Now, we already talked about what happens to the NADH in that electron transport chain when our final electron acceptor that's going to ultimately take the electron is oxygen producing water. But that's only when oxygen is present. When oxygen is not present or oxygen is not utilized, this NADH to NAD+ reaction still needs to occur, but how it's going to occur is slightly different. Under anaerobic conditions, which means without oxygen, NADH is reoxidized back by fermentation. There are many different types of fermentation, but they all operate to achieve the same goal, which is to regenerate that NAD+. In other words, to oxidize NADH back to NAD+. By removing the electron from NADH. The overall yield of ATP is only 2, the ATP that is made in glycolysis. So the NAD+ is needed for glycolysis. Without it, glycolysis will not proceed at all, and no ATP is going to be made. So let's look at some examples of fermentation. The first is called lactic acid fermentation. The end product is lactic acid, which is also known as lactate. And NADH is used to reduce pyruvate to lactic acid. Now, the goal of this is not to produce that lactic acid. The goal is to regenerate that NAD+. This occurs in many microorganisms and complex organisms, including invertebrate muscle during exercise when oxygen cannot be delivered to muscle fast enough. You see this very common in runners, and they get those lactic acid crystals build up in their muscles, which can be very painful. Typically, athletes will use rollers to help massage out the excess crystals. So real quickly, let's take a look at this process. We know that glucose in the process of glycolysis is broken down into two pyruvates. In this process, we are generating the ever-important ATP as a result. Now, in order for this reaction to take place and to produce that ATP, NAD plus is needed and will be reduced to NADH by oxidizing the breakdown products of glycolysis, of this glucose here. So without this step right here, this process will not proceed. So we need to have a constant supply of NAD plus. That way it can be available to be reduced to NADH. So what organisms do is they take that pyruvate and use an enzyme to convert it into lactic acid. By converting that pyruvate into lactic acid, it can take the electrons away from NADH, oxidizing it back to NAD+. Then the NAD+ can come back to glycolysis and allow glycolysis to continue to produce that ATP. Once again, the purpose of fermentation is not the end goal. It's not the end reaction here. This is not usable to many organisms. The goal of fermentation is to oxidize NADH back to NAD+ so that the NAD+ can come over and continue the pathway of glycolysis. That way the organism is able to phosphorylate this ATP through substrate level phosphorylation. Another type of fermentation is alcoholic fermentation. The end product is ethyl alcohol, which is also known as ethanol. Pyruvate is converted to acetaldehyde and CO2, carbon dioxide, is released. The NADH is used to reduce the acetaldehyde to alcohol, thereby regenerating that NAD+. More specifically, oxidizing NADH back to NAD+. This occurs in certain types of yeast and some plant cells under anaerobic conditions, again, which means without oxygen. So you can see this pathway looks similar to the lactic acid fermentation. We still have glycolysis breaking down glucose into pyruvate and producing ATP through substrate level phosphorylation. And in order for this process to occur, you need NAD+ present. That NAD+ is going to be reduced down to NADH by harvesting electrons away from the byproducts in this reaction here. Now, in order for this process to continue, we've got to take that NADH and we've got to remove those electrons and get it back to NAD+. So what these organisms will do is that they will take that pyruvate and with the help of several enzymes, they will convert that pyruvate into a molecule called ethanol. Now again, the end product or the end goal of this reaction is not to produce the ethanol. That's a waste product. Instead, the goal of this reaction here is to oxidize NADH back to NAD+. So the electrons are going to be transferred from NADH to this molecule right here that will eventually become this end byproduct. And that NADH is oxidized back to NAD+, where it can go back to glycolysis and allow glycolysis to continue. As long as there is NAD+ present, glycolysis will move forward and you will be producing ATP through substrate level phosphorylation. Metabolic pathways are linked. There is an interchange of molecules into and out of the pathways for synthesis and breakdown. Carbon skeletons, molecules with covalently linked carbon atoms, can enter catabolic or anabolic pathways, and these relationships comprise a metabolic system. What that means for us is that this complicated pathway that we've now finished and covered, starting with glycolysis, going to the oxidation of pyruvate, through the Krebs cycle, and the electron transport chain to produce ATP through oxidative phosphorylation can be done or can be completed even without the presence of glucose. You can have other types of molecules that can enter into these biochemical pathways at different points. For example, in the breakdown of lipids, a process known as beta oxidation, the byproducts can be entered in, such as glycerol or some of these fatty acids can enter in at the end of glycolysis or right after the oxidation of pyruvate. In the case of the breakdown of proteins, a process called deamination, several of the amino acids can enter in or be converted into products of the citric acid cycle. Some of the amino acids can even be brought in at the point during glycolysis. So these biochemical pathways that we covered in extensive detail are the same as any other biochemical pathway that can be entered at any point. And so in the process of proteins and lipids, and to some extent purines, which are the nucleic acids, can contribute to the production of energy. I hope this video was helpful to you all. Thank you so much for watching. Bye.